All right, welcome everyone to the MCHRI seminar series. We'll go through some slides about upcoming events while we're waiting on everyone to trickle in, and then we'll be able to get started. So next month on October 4th, we'll be having Dr. Pasca speak with us at our seminar series about using assembloids to investigate brain development and disease, which would be very interesting. So I encourage you all to come to that. And then in December, we have several members from our Global Child Health Program presenting for our series. And then, of course, on October 28th, we have our 4th annual Stanford MCHRI Symposium. I encourage you to all to please register and attend this event. It will be headlined by our keynote speaker, Dr. Moley, who is the Deputy Director of Reproductive Health Technologies at the Bill and Melinda Gates Foundation. And we'll be speaking on global priorities in reproductive health technology. We have 4 sessions full of really fantastic speakers doing amazing research. So again, I really encourage everyone to register for and attend this event. And this is just showing the members of the MCHRI Education Committee who are putting together this seminar series and the annual symposium for you. And after the talk today, you'll be receiving a link for an online survey. So we ask you to please fill out the survey and return it to us. And we'll wait just another minute for a few more people to join, and then I'll start my introduction of Dr. Prince. All right, so I think I will go ahead and start my introduction. And so we are very honored to have Dr. Prince here to speak with us today. He is the Division Chief for Neonatal and Developmental Medicine here at Stanford. He originally got his start at the University of Miami, receiving a Bachelor of Science in Chemistry. And as a UM grad myself, I have to say it's a great place to go to college. He then did his MD PhD at the University of Alabama at Birmingham with a focus in cell biology, and then additional scientific and clinical training at the University of Iowa, including his neonatal perinatal medicine fellowship. He spent some time at Vanderbilt University before becoming professor and chief of neonatology at UCSD and Rady Children's Hospital. As a physician scientist, Dr. Prince has a long and distinguished career leading a basic science laboratory focused on the mechanisms regulating developmental immunology and lung injury and repair, as well as, importantly, focusing on mentoring clinical and scientific trainees who have gone on to establish their own successful careers. Today, Dr. Prince will be discussing the role of lung macrophage populations in the pathogenesis of bronchopulmonary dysplasia and in lung injury repair, as well as on the molecular basis for newborn susceptibility to group B strep pneumonia, with the goal of developing this fundamental biology work into novel precision treatment strategies for newborn lung diseases. Welcome, Dr. Prince.
Thank you. I'm going to get my slides up here. Okay, I hope everybody can see that all right. Thanks for that nice introduction, and it's always great to talk about the old days with the fellow Miami Hurricane, so I appreciate that. We are interested in about why certain diseases appear to affect babies, and as a neonatologist, it's it's kind of obvious there. But there really are some things that are unique about the diseases that affect babies, and it's not always kind of exactly what we've always thought. So pneumonia is actually one of the major cause of death in children under 5 around the world. This has always seemed to surprise me when I would see this data, but it's consistent and it's clear. Now, this is kids all the way up to age 5. And if we look just at infant mortality, some of the things that we would probably know, like obviously prematurity is a major cause of infant mortality, birth asphyxia and injuries, congenital anomalies. But infections as a whole are actually the number one group, and largely within that group are pneumonia and sepsis. And while not every case of sepsis starts with pneumonia, many of them do. And so we've got this, very significant classification of diseases that are really devastating infants and young children around the world. And what's really kind of alarming is how little impact we've been able to make on these, even with in modern technology and antibiotics, et cetera. And so as we got started in some of this work many, many, many years ago, we found out a couple of things that really kind of changed the paradigm. And to me, the most important paradigm shift is this: babies are not weak. There's always been this kind of understanding or conventional wisdom that babies get certain diseases because they're small, they're immature, they're just not well developed. Well, it's not quite that simple. And babies are pretty tough. I mean, when they're born, their head gets, shaped into and basically like a large gourd or zucchini, and yet they come out and they can be neurologically normal within minutes. Their lungs go from being completely full of fluid to full of air within seconds. Their skin heals better than an adult's after an incision. They can eat every 3 hours, sometimes every hour and a half, and they can double their weight in 4 months. They can be on a ventilator for weeks, if not months, while not requiring extensive sedation. They can tolerate feeds. They can be held by their parents, look around, interact, all while being critically ill and on life support. And their immune system is amazing. In utero, they are showered constantly with antigens from their mom, including some of the maternal cells, and they have to decide which of these antigens do they make an immune response to or not, which ones do they tolerate. And then within literally a matter of seconds, they have to transition from this intolerant state to one that protect them against microbes that they encounter in the outside world, and then somehow decide, which of these microbes are beneficial to me and should I allow to colonize my skin or GI tract. And that in all this time trying to ward off severe pathogens. So there's an amazing amount of things that babies have to accomplish. And so the idea that they're weak and therefore susceptible to certain diseases, I think is a little bit misguided. So what we try to do in the lab is we try to always start with, well, what is it that we know? And then what is it that we don't know? And it's this path from the known to the unknown that we've been actually tackling. Now, it hasn't always been in a straight line, and it hasn't always been completely under control, and it certainly hasn't necessarily been always very efficient. But the idea is to take some of these things that we know on a global level, that we've observed over time, what do we know, and get it down to a specific molecule. And the idea being, can we understand disease pathogenesis better, and can we identify new approaches for therapeutic intervention that could save babies? So we know, let's start with pneumonia. So we know kids get pneumonia, and kids get pneumonia from a lot of the same pathogens that adults do in time to time. Most kids get viral pneumonia, but of the bacterial pathogens, they can get pneumococcal pneumonia and they can get H flu, which is hopefully thankfully a lot less common than it used to be. And we've known this for quite some time. Babies get a different type of pneumonia. They can be obviously much sicker from it, and they're more likely to develop septic shock and respiratory failure from it. But they're more likely to get pneumonia from bacteria such as group B strep, Listeria, and E. coli. And so this incidence of group B strep pneumonia in the newborn period was really intriguing to us. And there's a few things that we know. So group B strep is also known as streptococcus agalactiae. It was actually the name comes from the fact that when dairy cows would get mastitis from group B strep, their milk production would drop. And so it infects a number of different animals on our planet. It's the major neonatal pathogen. but it rarely causes disease in healthy adults. It can cause some infection in immunocompromised adults and very elderly, but it's extremely rare. And it's estimated that Group B strep causes around 150,000 deaths worldwide each year. And I'm still, every time I see that number, it's just amazing to me. And so, well, why is this? And why can't we do a better job in treating it? And so I was lucky that my colleague at UC San Diego, when we started this work, is Victor Nizet, a very good microbiologist in this area. And we started a collaboration. And a graduate student in my lab, Sean Lund, said, well, we first probably need an animal model of this disease. So he spent some time developing a Group B strep pneumonia model using both adult, juvenile, or day 7, and neonatal mice. And the idea being, if we could understand some of the mechanisms using mouse models, maybe we'd get a better idea of how to treat babies better. And using this model, he showed that only in neonatal mice, you saw about over 20% of the mice would die in the first few days of life. And what I don't show here is that if you take this time course out longer, you see a second wave of deaths at about 10 to 14 days only in the neonatal mice, not even in mice that are infected on day 2 of life. And it's that late onset of death is actually correlated with bacteremia and meningitis, similar to what we see in humans. So one possibility is that the adult and juvenile mice just aren't bothered by Group B strep at all when you put it in the lung. However, that wasn't the case. When we look at the pathology of the lung, 1 day after infection, all adult, juvenile, and neonatal mice all have signs of pneumonia and inflammation in the lung. There's influx of inflammatory cells, there's damage to the air spaces, there's atelectasis. All different stages have pneumonia at 1 day. However, a week later, things are very different. The adult lung is completely clear. Not only is the juvenile lung clear, it's actually undergone a degree of secondary septation, which is what we would expect during this period of development. However, the neonatal lung still looks like it has evidence of pneumonia. We still see inflammatory cells. We still see structural damage and injury to this lung. So while Group B strep can cause an acute pneumonia and inflammation at all stages of development, it causes a persistent lung pathology in neonates. And so our interest in lung immunity is kind of built around some of the work that we've done in alveolar macrophages. So this is an electron micrograph, colorized, yes, of an alveolar macrophage within the alveolar space. So these are the alveolar macrophages. They patrol all of the alveoli of the lung. And they're the key immune cell that's responsible for removing debris, killing pathogens, and actually recruiting other inflammatory cells if necessary. And they do this by expressing different inflammatory mediators and cytokines. And so Sean wanted to look and say, well, is there anything different about how these alveolar macrophages are functioning in the neonatal response to Group B strep versus, say, other stages of development? So one of the first things he did is he just took some pictures. He looked at lungs 24 hours after infection from adult mice or neonatal mice. And he stained the cells, the macrophages and Group B strep, and looked at them on the confocal microscope. So on the left image, that's an adult lung. The macrophages are stained with the antibody against CD68, and they're green. All the Group B strep is red. And you can see that a lot of the alveolar macrophages are taking up Group B strep bacteria. There's still some that hasn't been taken up yet, but the lung is doing a pretty good job of getting rid of the Group B strep, and when the macrophages take it up, they'll kill the bacteria. However, when we looked at the neonatal lung at the same time point, there are alveolar macrophages in here, and there are macrophages in the interstitium, and they seem to be trying to engage the red clumps of Group B strep, but they're not doing a very good job of taking it up. And so it appears that there's a defect in Group B strep phagocytosis in the neonatal alveolar macrophage. And that correlates with the killing of Group B strep. So these are CFU plots of the number of bacteria left in the lungs of adult, juvenile, and neonatal mice. And you can see on this log scale of CFUs, the adults are pretty good at getting rid of Group B strep over this 24-hour period. There's still some at 24 hours, but a lot of them have been cleared. The juvenile mice aren't bad. I mean, there is some clearing. There's still some bacteria there 24 hours later, but we really don't see any decrease in the number of GBS colony forming units in the neonatal mouse in the first 24 hours. So not only are the alveolar macrophage is not able to take up the group B strep, the lung's just not able to kill it. There's also a defect in the inflammatory response, and mainly the kinetics of the inflammatory response. In adult mice, they start to express inflammatory cytokines within a couple hours. So here we're looking at IL-1 beta, IL-6, and CXCL1. Cytokines and chemokines are kind of classic inflammatory mediators. And this is looking at gene expression within the lung. And you can see that they start to go up within 2 hours, and then by 24 hours, they're back down to baseline. In the neonate, we really don't see expression of these cytokines and chemokines until about 24 hours. So there's something about the initial immune response that's defective, as well as a defect in killing. Now, we did a lot of experiments to try to understand the mechanisms of this from the host side and from the pathogen side. And one of the virulence factors that we identified as key in this is the polysaccharide capsule of group B strep, which is a very large capsule, and it's completely coated in sialic acid as a terminal modification. And as a matter of fact, if you use group B strep that has a defect in the formation of this the sialic acid capsule, now neonatal alveolar macrophages can phagocytose the bacteria. And so here we've affected both adult and neonates with this mutant group B strep, which lacks sialic acid. And you can see that the adult macrophages will take it up. Also now we see neonatal macrophages can take it up. And this correlated also with the ability to kill and the resolution of pathology. Well, the way that you sense sialic acid is actually through a series of proteins on the cell surface called siglix. And these are lectin-like molecules that can bind sialic acid, and they can do a number of different functions. They can promote phagocytosis and inflammation, those listed over here on the left, or they can actually act to suppress the immune response through some inhibitory ITIM domains on their intracellular tails, and those are the siglix over here on the right. So we knew that siladhesin, which is also siglic 1, it was important for binding the group B strep polysaccharide capsule. And we also knew that alveolar macrophages express both siglic E and siglic F, siglic F coming on in mature cells, siglic E being present at birth. And these are both inhibitory siglix. And so we did a series of experiments to try to understand what is the balance of these expression of these different siglet molecules, and how might that contribute to group B strep pathogenesis in neonates. And one of the things that we did is we used mice that were defective in siglic E, which is an inhibitory siglet, and found that those alveolar macrophages are capable of taking up group B strep better than wild type macrophages. So the presence of siglic E at birth might actually act as an inhibitory factor in preventing the phagocytosis and killing of group B strep in newborns. And it's actually the lack of siladhesin in these cells at birth that makes them susceptible. And we did another series of experiments where we showed that even after 1 day, day 2 of life in mice, you start to see siladhesin expression, and now those mice are not susceptible to group B strep. So it's only right at birth that this susceptibility window occurs. And that's what we see in infants. It's probably group B strep that they are exposed to as they come through the birth canal or right around birth that makes them susceptible. The other interesting thing is that the two receptors together are able to achieve a balance in whether or not group B strep is killed and removed from the lung, or whether or not there's an inflammatory response. And so after doing a number of experiments, we came up with this model. Whereas the adult alveolar macrophages express both siglic E and siladhesin, group B strep can bind both of them, but that the overwhelming function is that siladhesin expression of group B strep is able to cause phagocytosis and killing, activate the immune response, and release cytokines, resulting in resolution of the pneumonia and infection. However, in the newborn, alveolar macrophages express siglic E, and so group B strep binds to that receptor, suppresses immune activation through the ITIM domains, but those cells don't express siladhesin yet. So therefore, there's no pro-phagocytic or killing or inflammatory response. And as a result, you get persistent infection. A few other experiments showed that group B strep through siglic E can actually suppress the expression of siladhesin, at least transiently, again, widening this window of susceptibility. So we're now doing a number of experiments trying to understand the differences, interactions between the host and the pathogen at this level, and including trying to understand, using a different selection of mouse models, what the role of cyclic E in this inhibitory function might be in utero. Is this part of the immune tolerance system that is important for newborns in utero, and what exactly does that do? Also, we're trying to understand, are there ways to increase the expression of sialadhesin in newborns around the time of birth to promote the ability of newborn alveolar macrophages to kill group B strep. So again, babies are not weak. There is a shift, a developmentally programmed shift in the expression of their receptors on their alveolar macrophages that appear to be responsible, excuse me, for the susceptibility to group B strep. It is not exactly an inability of their cells to mount a robust immune response. Rather, there's some sort of paradigm about which sialic acid molecules activate the immune response at that time. Well, another disease that we're very interested in is a disease that afflicts mainly preterm infants. And there's a preterm infant who's intubated on a high-frequency oscillating ventilator. And we know that infants that are born extremely premature, that have respiratory failure, are at increased risk of developing a disease called bronchopulmonary dysplasia. Here's a baby I took care of down in San Diego. This is Lexi. She was one of the smallest babies that we had down there. She was born weighing about 11 ounces, under 400 grams at birth. You can see she is intubated here on a ventilator. And even when she was older, right before she was going home, she was still on some supplemental oxygen. Excuse me. So she does have a diagnosis of bronchopulmonary dysplasia, or did at the time. She went home on oxygen, but was actually taken off oxygen after only about 3 months being home. And to this day, she's doing very well. She's a very active and fun preschooler. And so, but she did have a defect in her lung maturation as she went along her NICU course. And so bronchopulmonary dysplasia is a disease of abnormal development of the lung that only seems to affect babies born within a certain window of gestation. And so this is just a model of lung development. The lungs go through different stages of development, classified based on the way that they appear. This is lung development in mice. It's a little bit easier to get all the different stages here. Babies that are born extremely premature and at risk of bronchopulmonary dysplasia are born here in this cannulicular stage of lung development. And if you look at this lung here, it's amazing to think that this could actually function to exchange gas and oxygenate. However, it is, not without some consequences, however. And it's hopefully babies will be able to transition from a lung that looks like this over into the more saccular stage and alveolar stages of development, but it doesn't always happen that way. And bronchopulmonary dysplasia, which was first characterized here at Stanford back in the 1960s, is a disease of abnormal development. This is what it looks like on X-ray. You get these cystic, kind of heterogeneous looking lungs. This is what it looks like under pathology. This is a patient, unfortunately, I took care of who didn't survive, and we have autopsy pathology here. This to me looks like a very large cannulicular stage lung. It gets bigger, but it just fails to develop. And so what we see in the pathology of these children with bronchopulmonary dysplasia is that their lungs have a thickened interstitium. They have these kind of larger air sacs, if you will. They lack normal alveoli, and actually this was thought to be an emphysematous bleb in a patient. It's actually just a very under alveolarized area of the lung. They also have very abnormal capillaries throughout their lung. And some of these capillaries are actually dilated and run through the interstitium and may function as an alveolar venous shunt going through the lung as well. And so BPD is actually pretty common. It only affects preterm infants. It's a disease of arrested lung development, and it correlates with infection. And that's what I'm going to talk most about. But because of the high rate of prematurity in the US, we have a high rate of BPD, and the estimates every year are somewhere between 10 and 15,000 new cases each year in the United States. So because it's related to infection and inflammation, and those clinical connections have always been observed, we decided we were going to try to understand the mechanisms of how infection and inflammation could affect lung development at early stages of lung development. Now, when we first started these experiments, and this was, 15, 20 years ago, maybe about 15 years ago, I guess, there wasn't much known about macrophages in fetal tissues, period, and especially in the fetal lung. So Reet Vandermeer in my lab did an experiment. She took some embryonal mouse lungs and she stained them for macrophages. And the red cells here are the epithelial cells, so they're the airways, and the green cells are macrophages. And you can see there are a lot of macrophages in the fetal lung. Now we know that, of course. There's been a lot of work done since this initial experiment in tracking the formation of macrophages from various precursors and how they populate all the different tissues of the developing embryo and fetus and actually newborn. But what we didn't know yet is how functional were they. So Ashley Hipp-Stouch, a graduate student in my lab in collaboration with Tim Blackwell's group, took advantage of a mouse model where any cell in the mouse where there was NF-kappa B activation, which is the major pathway of innate immunity, any cell in that mouse where there was innate immune activation would express the green fluorescent protein from jellyfish. So we could actually look under the microscope and identify which cells were responding to inflammatory stimuli looking for this green fluorescent protein. So these are explants of fetal lung tissue treated with E. coli endotoxin lipopolysaccharide, or LPS. And within 2 hours, you can see these green cells in the fetal lung explant. And these cells are macrophages. So they're stained with CD68 and they're green from GFP. So the yellow is the sign of co-staining. Now, there are some macrophages that aren't expressing green fluorescent protein, and we didn't know what that meant for a long time. But what's interesting, so 2 hours, you see that the macrophages are green, but then later the other cells in the lung become green. And that to us suggested that the macrophages were the initial site of immune response. They are making substances that then activate immune signaling in adjacent cells within the lung. And so we did a series of experiments to try to understand how this might affect development. But like I said, work has been done since those initial experiments to really trace out where macrophages come from. The first wave of macrophages come from the early embryonic yolk sac, and they show up in the embryonal lung in mice at E10, so very early. Basically, the first day that there's a lung, there are macrophages from the yolk sac showing up. But then there's also a wave of macrophage or hematopoietic precursors that go to the fetal liver. And a few days later in mice, these cells in the liver give rise to a number of different cells throughout the blood lineages, including a second wave of macrophages. They're actually monocytes when they leave the liver that also go to tissues, including the lung. So by embryonal day 14 in a mouse, and again, embryonal day 20 or 21 is term in a mouse, you have two populations of macrophages, one that comes from the yolk sac, one that comes from the fetal liver. And these carry slightly different marker expressions. So the cells from the yolk sac, so embryonal day 13, this is from the lung. Embryonal day 13 in a mouse, you only have cells from the yolk sac, and they're F480 high, and they express low levels of CD11B. However, the next day, you start to see cells that are CD11B high and F480 low, and these persist. And so these are the cells that come from the fetal liver. So you have two populations of macrophages that show up in the lung. And it's these CD11B high monocytes from the fetal liver that eventually turn into alveolar macrophages. And we'll talk about that a little bit later. But through a series of experiments, we figured out the role of macrophages in bronchopulmonary dysplasia. So the fetal lung that does contain macrophages does respond to infection and inflammation. And there's NF-kappa B activation within these macrophages, and that leads to a number of different things. It causes recruitment for additional macrophages and monocytes from the circulation. It causes the macrophages within the lung to actually proliferate. And now we know more and more that alveolar macrophages especially can renew themselves over a lifetime. They do not need continuous supply from the circulation. Ashley also showed that these macrophages are more likely to accelerate their maturation if they were exposed to early inflammation or infection. And they make a number of inflammatories. The major one that we think is important is IL-1 beta, that then target gene expression in other cell populations within the lung and cause abnormal development. So when macrophage IL-1 beta signals on the mesenchymal cells, or the cells that will become fibroblasts in the lung, it causes them to make less FGF10, which is important for lung development. They make less alpha-8 integrin, which is also important, and a number of other developmental genes that are affected by this early exposure to inflammation. This is stage specific. It does not do the same thing in a term kid who's exposed to pneumonia as well. It's only during this stage of development. So we thought we figured out a lot of these things and found some potential therapeutic targets. However, there were a lot of questions that we still didn't answer. So why were the effects of inflammation so stage specific? And why doesn't every macrophage respond similarly? You know, only a subset of those macrophages were responding to LPS when we studied it. How does normal development then control inflammation? Because I'll get into it a little bit later, that the inflammatory response appears to be a little bit affected by developmental stage. Do newborn macrophages mount a normal immune response? You know, again, are babies truly weak, or is their immune response actually normal? And what does this have to do with disease? So we did a few more experiments. So Omar Lakhdari in my lab took advantage of the fact that this marker expression allows him to separate the cells in the fetal lung and in the fetal liver that come from the yolk sac or the liver itself. And he showed that there's a difference in how they respond. And so he isolated these macrophages from the two different populations, the F480 high macrophages from the yolk sac, the CD11B high macrophages from the liver, isolated these cells both from liver and lung, and tested their response to E. coli lipopolysaccharide. And what was interesting, it was only the CD11B high cells from the fetal liver that expressed IL-1 beta or IL-6 and that responded to LPS. The cells from the yolk sac appeared to be unresponsive. And Katie Anderson, who is a neonatology fellow working in my lab, showed that they were also unresponsive to every other toll-like receptor ligand that we tested. So that there's clearly a difference in the immune response in these two different macrophage populations. They're also different in how they are able to form an inflammasome. And an inflammasome is a cluster of proteins that actually is responsible for the cleavage and release of IL-1 beta. And two of the key parts of the inflammasome are NLRP3 and ASC. And here we're looking at an F480 high macrophage, which is a yolk sac-derived macrophage. The expression is there, it's kind of low. And when you add LPS, you start to see a little bit of yellow. Those are inflammasomes coming together from the two different proteins. However, when you look at the CD11B high cells, and this is all from fetal lung, the expression, at least of NLRP3, is a little bit higher. And then when you treat these cells with LPS, you see a lot more yellow. Almost the entire cytoplasm is full of inflammasome assembly. So again, showing that these two different populations of macrophages have very different immune functions. And so when we go back to this idea that macrophages that come from the fetal liver then traffic to the lung, these are pro-inflammatory, or at least more capacity to be inflammatory. What's interesting is these are the cells that will eventually become alveolar macrophages. And they don't differentiate into alveolar macrophages until sometime after birth. In mice, it's usually about day 3 or 4 after delivery. In humans, we honestly don't know, but we think it's probably in the several days after delivery. So Asami Honda in my lab, who's a graduate student who just defended actually last year, she took this heroic RNA sequencing project on of sorted macrophage populations throughout development. And she compared neonatal alveolar macrophages from day 4 mice to adult alveolar macrophages. And they both express all the genes that make them an alveolar macrophage. So these are some of the classic alveolar macrophage signature genes. However, the neonatal alveolar macrophages still retain some of their monocyte markers. So these are kind of thought to be classic monocyte signature genes, but they're still present within alveolar macrophages in the neonatal lung. There's a number of genes that were different. And it's interesting, the adult alveolar macrophages express a lot more genes involved in lipid signaling, PPAR gamma signaling, which we know PPAR gamma is important for alveolar macrophage differentiation. But the neonatal macrophages expressed a lot of genes involved in the inflammatory response. So again, suggesting that these cells, even in a newborn mouse, had this pro-inflammatory phenotype, if you will, compared to an adult macrophage. And so here are some of the genes. Some of the genes that are higher in adult alveolar macrophages had to do with lipid signaling and transport. And a lot of the genes that are higher in neonates have to do with inflammation. So you have 3 inflammatory marker genes that are higher in alveolar macrophages for neonates. And again, these are under control conditions, and including a PDGF beta, which is a growth factor important for development of a number of tissues. So they still have also maybe some expression of genes important for morphogenesis. Asami did an experiment to try to test out the kinetics of this, and what she focused on was just interleukin-1 beta, IL-1 beta, to see, well, how does this change over time around delivery? And it was built on some work Ashley had done looking at a lot of different inflammatory genes, and we always saw this peak right around the time of birth, and we never really knew what it meant. So Asami focused on macrophage populations, and it's interesting, even though those fetal monocytes express IL-1, they're here, but they're a lot lower than what is going to happen over the next several days. The fetal monocytes, as they go into the lung 3 days later, express a lot more IL-1, and some of them are already differentiating into prealveolar macrophages. Right at birth, you still have some fetal monocytes and these prealveolar macrophages, and they have a lot higher expression of IL-1. As cells differentiate into alveolar macrophages, really mature cells, then the IL-1 expression starts to fall. So there's this maybe potentially a priming of the inflammatory immune response happens in utero. Right around the time of birth, it's maximized. And then in the adult and during development, then that inflammatory response is then attenuated over time. And so exactly what's controlling this and what this means for immunity, we still don't know. But what's interesting is that Asami showed that the macrophages, the alveolar macrophages in neonates and adults use a very, very different transcriptional mechanism, set of transcriptional mechanisms. She did a lot of attack-seq work to look at areas of the genome that were open so that those genes could be expressed, and found that within those different open chromatin regions, adult macrophages were more likely to use transcription factors as part of the KLF family member, the nuclear receptor family, there's PPAR gamma, NR3C1 is the glucocorticoid receptor, and members of the STAT family. However, neonates were more likely to use motifs and enhancers that would bind AP1 and NF-kappa B family members, and these are a classic innate immune response transcription factors. So very different chromatin landscape in neonatal and adult macrophages, and that contributes to the differences in gene expression. She next tested whether or not the response was different. So what she did is she treated day 4 neonatal mice and adult mice with intrapulmonary LPS. And then she looked to see which cells were responding. And here she's looking at the expression of pro-IL-1 beta. And in adults, under control conditions, there's really no alveolar macrophages expressing IL-1 beta. You give them LPS and most of them do. So that's classic. And there's also a few cells within the interstitium, or interstitial macrophages or monocytes, that will express IL-1 beta in the 2-hour time course of this experiment. Similar, we saw the same kind of response in newborns. Now, again, there's already a lot more cells that are expressing IL-1 beta in the newborn under control conditions. But when you add LPS, you get about the same percentage that become positive, both in the alveolar macrophage and the interstitial macrophage compartment. So the response to LPS in vivo was very similar between newborns and adults. The genes that they express after LPS treatment are also very similar. The same types of inflammatory mediators are expressed in adults and neonates when you give them bacterial challenge. We really didn't see much difference here. The only difference that we saw was in the fold change of the inflammatory mediators. And it wasn't because the neonatal mice weren't able to express these inflammatory genes. The difference was because they already expressed them under baseline conditions. So the amount of increase was a little bit less than compared to adults. The genes that were turned on after bacterial stimulation, or at least endotoxin stimulation, were almost exactly the same across the board. They used the same transcription factors. Here are the motifs that were enriched. And the expression of the different transcription factors and the motifs that they recognized within enhancer regions was also very, very, very similar. Okay, so that's in mice. Now, what might this have to do with bronchopulmonary dysplasia in kids? So we did an experiment, a series of experiments, where we were isolating tracheal aspirate macrophages from premature babies on the ventilator and asking to see if there was gene expression in those macrophages correlating with disease outcomes. So even when we isolated cells on day 1 of life, we saw differences from the kids who went on to develop bronchopulmonary dysplasia many months later and those who didn't. And a lot of the differences were inflammatory genes. These chemokines and cytokines that we measured that were higher in the kids who eventually went on to develop bronchopulmonary dysplasia. Now, granted, it's like a lot of biomarker studies, there is some overlap, and we're talking about statistical differences, not necessarily a very, very clean predictive value of any one factor. We saw even more dramatic changes a week later, where different sets of cytokines and chemokines, including now IL-1 alpha and IL-1 beta, were higher in the kids that went on, in this case, developed severe bronchopulmonary dysplasia. So the macrophages in newborn infants and preterm infants that are pro-inflammatory, if you will, that inflammation in that period does correlate with the development of more severe disease. The other thing that we saw, though, was kind of interesting, was that there was a pattern of disease resilience in the kids that were on the ventilator for 3 weeks of life, but did not develop bronchopulmonary dysplasia or only had mild disease. So these were the kids that were off oxygen and off all respiratory support by 28 days of life. And so one of the first things that caught our eye was this gene MRC1, which is it's a mannose receptor, and it's actually acts as a scavenger receptor on macrophages. It's a lot higher on day 7 of life in day 14 and 21 than it was on day 1. And in the kids who developed none or mild BPD. The kids with severe BPD, it was more scattered and didn't see the same clear pattern. We saw a very similar pattern looking at other scavenger receptors on macrophages, Marco and MSR1, whereas in the kids who didn't develop severe disease, the expression went up pretty dramatically in the first week of life and then stayed high. Similar patterns with fatty acid binding protein 4 and the complement protein C1q alpha or C1qa and C1qb. Interestingly, C1q's family members are highly expressed on those yolk sac macrophages that are relatively unresponsive to endotoxin. So Debasha Sahu down in San Diego helped us do this in a much more rigorous way. We looked at all of the different data sets that we had, and we had microarrays on, we had about 220 microarrays from these patients over time, and found different patterns, genes that would go up between day 1 and day 7 and stay high, genes that would come up later, and then genes that would go down between day 1 and day 7. And these are those patterns and clusters in kids with severe BPD, and this is the patterns that we saw in kids with no BPD or mild disease. And the interesting thing is that kids with no disease or mild disease had a much bigger group of genes that went up between day 1 and day 7 and stayed high. If you take these same genes from the kids that were quote unquote resilient to disease and looked at the expression of those genes in the kids who developed severe BPD, they're all low and they stay low. So we think that this pattern might actually be predicting a group of genes that are important for macrophage development and disease resilience. And here we looked at some of the transcription factor motifs that were important for the expression of those genes. Again, NF-kappa B being one of the ones that showed up in the kids with severe bronchopulmonary dysplasia. So again, the path hasn't always been very smooth or straight. But the kind of the take home is that when infants actually have to transition from a state of immune tolerance to immune activation, and that transition brings in a whole bunch of possibilities of susceptibility to disease. The pathogens have learned, especially group B strep, to exploit these specific opportunities. Newborn macrophages are actually inflammatory. They are primed or just maybe constitutively activated for reasons we don't completely understand. However, their innate immune response is very similar to adults. When we actually looked at what they do when you stimulate them, the expression is the same as what you'd see in a more mature cell. And then finally, that this activation, it does affect normal development. And we now know that infection and sepsis is an increased risk factor for bronchopulmonary dysplasia. It has been known for a long time that infection, even in older children, not just extremely preterm, is a risk factor for cerebral palsy, and it may play a role in other disorders of normal human development. So again, babies are not weak. I think it's inherent upon us to understand that there are very, very unique molecular and cellular mechanisms that are going on within our patients around the time of birth that we need to understand better. And it's not appropriate just to say that they're lacking an immune system or they're deficient or they're whatever it is. No, they're different. And we need to understand the differences of that stage of development, just like we would want to understand the differences of any other stage of development. So I have a lot of people to thank. Here are the new members of my lab here at Stanford, past members of the lab, and a lot of fantastic collaborators that have helped us along the way. I was telling Melissa earlier, not only did I not study immunology early on in my education, I didn't really pay attention that much. And so as we got interested in disease mechanisms, I really had to kind of learn along the way and collaborate with a lot of fantastic immunologists that helped us out. The research was funded by the NIH and the Gerber Foundation. And I put these are two recent photos of our lab over the last number of years. And as I put them up there, I realized that, I've never actually taken a role in having quotas or trying to engineer the demographics of my research team. But especially now being in September and this Women in Medicine Month, I'm very proud of the fact that we've had a number of really brilliant, talented women who have worked in my lab, and that we've had a number of people from underrepresented populations that have done exceptional science. And it's really been a pleasure to have them as part of the team. So with that, I'll stop and be happy to answer any questions.
Thank you, Dr. Prince. That was a really, really fascinating presentation and really interesting work that you are doing. I'll start off with a question I really want to know about while people are getting their questions into the Q&A. I know that moms who start to go into preterm labor are given steroids for lung maturation. And if I recall from my early medical training and things, I thought that was related to trying to get more surfactant production. But I was wondering what impact that may have on this pro-inflammatory phenotype of these macrophages around the time of preterm birth.
Yeah, thanks for that. And so, right, I mean, almost every mom now in preterm labor gets steroids before the babies are born. And it does increase surfactant maturation. And I think it's probably got to affect to some degree, the inflammatory phenotype that we see in alveolar macrophages. We don't yet know how much. It's kind of hard to do those get control samples for that. And we're still trying to, understand that better, but it's got to have some influence. And the question is whether or not it's sustained and whether or not it's protective or not, that we just don't know.
Yeah. I was wondering too, in one of your very first slides, you showed that when talking about the GBS, you showed that the newborn or the preterm infant cytokine production was delayed, but it looked higher in the end. And then I know you were talking more about the bronchopulmonary dysplasia when you were talking about these hyperinflammatory macrophages, but I wonder if some of the outcomes you see with the GBS infections is related more to these delayed but hyperinflammatory responses from these macrophages rather than the inability to clear the bacteria itself.
Yeah, I think that's very possible. So, not only are the bacteria staying around where they can continue to activate, other mechanisms, through TLR receptors, whatever, but that actually very well could contribute to the mechanism. We know that adult alveolar macrophages are really good at suppressing inflammation after they mount an immune response. So if you could follow one bacterium coming into an adult lung, the macrophages would take it up, they could kill it, and they could suppress any immune response really very quickly. And that ability to suppress it in inflammation when it's not needed is something that's more prevalent in adults than in neonates. We don't know exactly why. It could be that the inherent nature of the alveolar macrophage probably has something to do with it. There's also probably fewer cells in the neonatal lung that can contribute to immune suppression and tolerance, like T helper cells and other cell populations that can help regulate that. But yeah, that was a nice observation, and it probably does play a role.
And our alveolar macrophages, can you subcategorize them into like M2 and M1 phenotypes? Or is that...
Thank you for asking that. So we started out with the project trying to do that and see if this was a difference between M1 and M2 polarization. And it turned out that paradigm really didn't apply very well to alveolar macrophages, because the same macrophage could share it with M1 and M2 markers. And while more mature alveolar macrophages do express more M2 markers, they're also very good at mounting a robust immune response, in seconds. And so there's just too much overlap, I think, in the lung to be able to make that kind of clear type of polarization. Having said that, there are alveolar macrophages that are more engaged within the lung epithelium and have a different phenotype than the ones kind of just rolling around, if you will. And then within the interstitium of the lung, there are probably at least 3 different populations of macrophages that have different relative expression of MHC class 2 and other molecules that are probably important for surveilling the lung. And there's even another class of macrophages that seem to hang out in the lung vasculature and are always patrolling the endothelium there. So a lot of different subcategories, but not as simple as the M1, M2 paradigm.
Okay. Yeah, thank you. We do have a question in the Q&A that I think it's that how, I'll get into the specific question, but I think the overall gist is how long do these early issues impact lifelong health for premature infants? So, for example, one of the questions says, I was born 6 weeks early. I have experienced, very bad case of strep, staph, and mono at age 16, chronic fatigue for 3 years at age 35, and chronic dizziness at age 65. Are these later diseases related to that prematurity, or how does that impact later life?
Yeah, we don't know all the answers to that. I'll be honest, we used to think that kids that were born after 32 weeks were probably fine because they weren't critically ill often, and they seemed to be pretty good when they went home. And now we're learning that they do have some issues around imperfect development, and they can have neurodevelopmental issues. We don't yet know exactly what the differences might be in their GI tract or their lungs and how that maturation occurs. One of the things that we're looking at in the lab now is the effects of early exposures and epigenetic changes that persist throughout life. And there may very well be some, and we owe it to the patients that we care for to know this so that we can, in case someone actually does develop lifelong problems, is it due to what they experienced around the time of birth, and how can we help them with that? Bronchopulmonary dysplasia does persist into adulthood. We're very fortunate that the majority of the kids do better with time, but we know that their pulmonary function tests are very abnormal when they're older, and they probably are. We know that they're at increased risk of pulmonary hypertension, and hopefully they never smoke, but there are going to be some issues around other exposures throughout their life course.
Great, thank you. We had another question come in. Are there ways we could nudge babies born with the higher risk gene expression patterns away from bronchopulmonary dysplasia, like drug targets or things like
yeah, that's exactly the approach that we're trying to do. His question is, which drug, which target, how do we do it? How do we deliver it? What cell do we need to target? When and for how long? And we just have a lot to figure out. I'll be honest, my lab has really been focused on identifying the targets. And we think we've found a couple, but that are important, but we haven't yet figured out how to manipulate them. So we got a little work to do.
Yeah, that's great though that you have some early targets that you might be able to work on. We had another question come in, and I know you already talked about later in life infections and things, but this one was specifically wondering about how the information can be connected to SARS-CoV-2 infections. Would infants be more vulnerable for these reasons, or as I was alluding to, I wonder whether it may have an impact later in life for that specifically as well.
So we feel really fortunate that neonates do not appear to be as susceptible to SARS-CoV-2 as other viruses or as older children and adults are. And one hypothesis, and there's some data from colleagues of mine have shown that they do express lower levels of ACE2 and the receptors for SARS-CoV-2 and in the neonatal lung, and that might be why they are less susceptible. Alternatively, it could be that it's all due to the secondary innate immune response, the interferon-mediated response, the vascular response that is isn't as robust in neonates or smaller children than in adults. It's still kind of a hypothesis. I think it seems to change by the day, but I think we did dodge a bullet. That's different than what we see with, say, RSV, right? So, babies are very susceptible to RSV, and they're also susceptible to flu. And so, we'll just have to kind of wait and see. And again, hopefully we just dodged a bullet there. But I think what we can learn about the response to all these different respiratory pathogens is the different types of innate immune responses and adaptive immune responses that happen and how they interact with the repair mechanisms in the lung. And so, even in the adults that are getting really bad COVID, it's not the initial viremia or the viral infection in the lung that causes most of the pathology. It's the subsequent inflammatory response and the inability of the lung to repair. And every one of these quote unquote cytokine storm models are a little bit different depending on which cells are involved and which profiles of cytokines and chemokines they're expressing and what sort of damage occurs. So it's going to be interesting to profile all of these with all the different cellular compartments in context so that we can develop more intelligent therapies, I think.
Great, thank you. There is one other question in the Q&A, but I think you already answered it when you're answering one of the very first questions about, later in life. The original question I asked was, someone who had other medical issues later in life. And the second question is from someone who had the opposite. They were very sick as an infant, but then did okay later in life. And so, I think you already got to that, where there's, so much else that plays into that it's hard to say.
Yeah, and that's the thing that we really need to focus on most, right? Is understanding, the precision health aspect of this and disease resilience. How is it that we have some babies who are born at 22, 24 weeks, under 500 grams, who are doing exceptionally well later in life? What is it that they make? What is it that they have? What are their cells expressing? Can we bottle it and give it to everybody? It's just really trying to understand the precision health aspect of this, knowing that every baby is different. They go through a different course. And can we understand that at a molecular level? And that's what we'd love to do. Yeah,
that would be great. All right, I don't see any other questions in the Q&A unless somebody gets one in right now under the buzzer. But while we're just giving them a minute to do that, I just want to remind as well, first of all, say thank you very much to Dr. Prince for being here with us today and giving such a fantastic presentation, and see if there is, and just remind everyone to please be sure to register for the symposium and attend, because that will be really fantastic. Yeah, I think the only things that have come in are sort of more details about the personal experience of the person who did well later in life. So I think we've already addressed that. So I think I will just say thank you to everyone for attending today as well. And we hope to see you next month as well. Thank you.